morning students. We'll continue with the topic plant growth and development. In the previous class we have gone through the two promoters, that is the auxins and what are the effects of the auxins and what are their physiological roles with their discoveries. Auxins and the gibberellins. Now today's hormone we will go with the physiological role of the cytokinins, how they were discovered and what are those effects, what are the physiological roles of these cytokinins. So this hormone cytokinins was discovered by the work of a teamwork of the scientist. The scientists who discovered this hormone were Skoog and Miller. Around the year 1955. They were working on the plant tissue culture, that is tobacco, on tobacco plant culture. So Skoog and Miller did several experiments in order to develop the callus. This callus is nothing but a mass of undifferentiated tissue. Callus is the mass of undifferentiated tissue. I like to give an external information on the process of tissue culture. In tissue culture, the concept of totipotency is being employed, where from any part of a plant can develop into a new plant. So in tissue cultures, the microshoots, that is the plants, are allowed to grow in a nutrient medium. You might have seen in the flask or in a larger size test tube, where the plants are being grown. They use a solution or they prepare a nutrient medium. Where a small piece of a leaf or a stem or the pollen grains, the male or the female gametified, is being added on this nutrient medium, that is in the flask or in the test tube. Growing of the microshoots in the nutrient medium, that process of growing those plants is called as a tissue culture. So they were culturing on tobacco plants. Yes, so the nutrient medium, this medium was being prepared and this medium contained all the necessary components or substances which was responsible for the formation of the microshoots. Before the microshoots, it will develop to form a mass of cells. This mass of cell is called as the callus. So here the nutrient medium is being prepared. This nutrient medium is prepared using the extracts. Then this nutrient medium contains extracts of vascular substance, yeast extract and coconut milk. So they prepare a medium. That medium contains the extract of the vascular substances or the yeast extract or the coconut milk, which induces the part to develop the group of new cells. That is the callus, or we can say as mass of undifferentiated widely used in the process of tissue culture. Even after series of experiments, they were not satisfied with the growth of the callus. Yes, so apart from this, in the tissue culture, the medium contains agar-agar and the starch and the potato starch, water and other antibiotics in order to stop the growth of the bacteria or the fungus. So that medium that was being prepared and it initiated the growth of the callus. With series of experiments, they found out that they were not satisfactory. They did not give satisfactory result for the growth of the callus. So they were disappointed with the fact that the callus was not growing properly. Then one of that, in that scoop, he added old extract of a DNA. An old extract of DNA was being added to the medium. When it was being identified and it was being studied, after studying it, it was being observed that there was growth of the callus. Growth of the callus. Now, this extract of DNA, then they could observe it, or they got an identification, or they made an study of this extract. So this old extract of DNA, which was responsible for the growth of the callus, that was being identified and it was named as kinetin. It was named as kinetin, which is a product of, it is a product of adenine, that is a purine and a component of nucleic acid. When they studied, when they observed that this DNA extract was responsible for the growth of the callus, that was chemically identified. That chemically identified was named as kinetin, and this kinetin is the product of adenine, one of the form of a purine in the component of the nucleic acids. The DNA nucleic acids are 4 types, that is adenine, guanine, thymine and cytosine. In that, kinetin contains the chemical component adenine, and this adenine stimulated the cells to divide, especially the cytoplasmic cells. So it was named as cytokinins. But this kinetin is not present naturally in the plants. So there was development of the calyx, that this chemical was identified, or it was being extracted from the dividing cells or the dividing regions of the plant cells, but this kinetin was not naturally present in the plants, but it was extracted as a component called as zeatin. So this zeatin was extracted further from the corn kernels and coconut milk, which was chemically studied and was identified as 6 furfuryl amino it was identified as a chemically it is known as 6 furfuryl of discovery of this cytokinins. It was discovered by Cook and Miller in around 1955. They were working in a teamwork of scientists. They were working on the tissue culture technique on the plant tobacco. So already in tissue culture we use the medium called as nutrient medium. So this nutrient medium contains all the necessary substance which is responsible for the growth of the plant. So if a small piece of a stem or a leaf is placed on the nutrient medium, with the help of the nutrients there is a growth of the callus, and that callus is nothing but the mass of undifferentiated tissue, where you will see a mass of cells on the medium. So this nutrient medium is prepared by obtaining the extracts of the vascular substances and even the extract of the yeast and coconut milk, which worked as that substances that stimulated the cells to divide. Even after doing many experiments, they were not happy with the growth of the callus. So here the Miller added old extract of DNA. When it was being added as a DNA component, extract of DNA, there was remarkable growth or rapid growth of the callus. So this chemical that induced the growth was identified as a promoter, and when it was chemically studied, it was called as kinetin. And this kinetin is the product of adenine, that is a purine, a component of nucleic acids. So already the nucleic acids in that adenine, it is one of the component of the DNA is the adenine, that is a purine base, So when it was chemically studied, and what being identified as a growth promoter. Here it was, this zeatin was not present naturally in the plants, but was being extracted from the growing tissues, growing or dividing regions of the plants in the form of zeatin. And the zeatin was being extracted from the corn kernels and from the coconut milk. Later on, it was chemically identified, and the chemical name of the kinins was called, what is 6-sulfuride-aminopurine, which was responsible for the growth of cytoplasmic growth, and it was named as cytokinins. Next, we will go with the physiological roles of cytokinins. What are the physiological roles? It is one of the cell dividing factor. It is a cell dividing factor which induced cell division and cell enlargement in the growing regions, that is shoot and root apex. discovered, what are its physiological roles was being identified, and how it induces or how it promotes the growth was being studied. So it was being identified as one of the cell dividing factors. As a factor, as a growth promoter, it induced the process of cell division, which made the cells to divide rapidly or very fast. Yes, especially in the apical buds and the meristematic cells. So this cytokinin, when was being treated, or the plants were being treated with the cytokinins, it helped in large cell enlargement and also in the cell division, especially in the growing regions, which are nothing but the shoot and the root apex, the tip of the roots and the shoots. So here the plant in tissue culture, if leaf is being used with the treatment with the cytokinins, it will develop to form, it will make the cells to divide very fast. So it is recognized as one of the cell dividing factors which induced the process, the cell division, especially in the meristematic cells. Then it also helps to produce new leaves, chloroplast of leaves, and formation of lateral shoots, lateral and adventitious shoots. So this cytokinins, when was being used as a promoter, it produced new cells. It produced new cells or of the leaves. It helped to develop new leaves in the plants and also help in the development of the chloroplast. Chloroplast was developed in the leaves and also helped in the formation of laterals and adventitious shoots. Already, what are adventitious? Those which grow from any part of a plant apart from the radicle. So you can see that even some shoots that grow from the branches, those growth of laterals and adventitious was being induced by this hormone, that is cytokinins. So if you take a developing plant, when the plant is developing or in the state of growth, so it helped to produce the new leaves very fast, and those leaves could produce large number of chloroplasts in the leaves, which helped them to carry out the other processes, physiological processes of the plants like photosynthesis and respirations, and help in the formation of laterals and adventitious shoots. Then it helps to overcome apical dominance and thus promotes the growth of axillary buds. So it helps to overcome apical dominance. Apical dominance, if there are apical buds, they will not allow the axillary buds to grow. Yes, that phenomena where the apical are dominant they are dominant over the axillary buds. So that phenomena is called as apical dominance, as we have studied in the Gibberellins part. So here, if you apply the cytokinins to the plants during the process of agricultural or horticultural processes, it helps to overcome the apical dominance, or it will initiate the axillary buds to grow, and thus it is initiating the growth of the axillary buds. In that way, it is overcoming the phenomena of apical dominance. Then it promotes nutrient mobilization and thus delay senescence. It promotes nutrient mobilization, then delay the process of senescence. The leaves, when there is a process of yellowing of the leaves, and that yellowing of the leaves is due to the loss of the chloroplast. So if the leaf loses the chloroplast, that the nutrients are being lost, or we can say that there is a loss of chloroplast, and this loss of chloroplast is due to the breakdown of the proteins that are present in the particular organ. So when there is a breakdown of such proteins, that protein's destruction will also destroy the chloroplast, and for that the leaves are yellow in color or become yellow in color. But if if the plants are being treated with cytokinins, we can avoid by mobilizing the nutrients, by transportation of nutrients to the leaves, and thus delay the senescence. Senescence means the process of aging. The process of aging is nothing but the yellowing of the leaves. So by treating with cytokinins, we can mobilize the nutrients and postpone the process of yellowing of the leaves. That means we can delay the aging of the leaves, that is the senescence. So this hormone, it promotes the nutrient mobilization and thus delay the process of the senescence, that is the process of aging. And it has the ability to bring morphogenetic changes in the callus and thus promotes development of micro shoots. This cytokinin, when it is added in the nutrient medium, it can bring the morphological changes in the callus, and that which there was when there is morphological changes in the callus, that will help in the development of the micro shoots, that is the shoots. So when it is being treated, there is a growth of the callus. This callus, if it is treated with cytokinin, you will find that there is a growth of micro shoots in the tissue culture technique. So it has the ability, the cytokinins have the ability to bring the morphological changes or morphogenetic changes in the callus and thus promotes the development of the microshoots. So once again we will go with the physiological roles of cytokinins. So it is a cell dividing factor. So it helps with the process of cell division. As I said earlier that it is present in those parts which are under the process of rapid cell division. So it is a cell dividing factor which induces the cell division and the cell enlargement, especially in the growing regions or in the dividing cell portions or the part where there is rapid growth, that is nothing but the shoot and the root apex. Then it helps to produce, when the plant is germinating, it will germinate to produce new organelles. So it is producing, it will help to produce new leaves and the chloroplast of the leaves. Not only that, it also helps in the formation of the laterals and the adventitious shoots. Then it will help to overcome the apical dominance. This apical dominance will induce the growth of axillary buds and thus promote the growth of axillary buds. Then it promotes the nutrient mobilization. The nutrients are transported to the regions of the leaves and it will prevent or it will delay the process of aging of the leaves. Then it has the ability to bring morphogenetic changes in the callus, but there is a rapid growth of the callus. This rapid growth of callus help in the development or in the most rapid development of the microshoots. So these are all the physiological roles of the cytokinins. Then we will go with the inhibitor hormone. That inhibitor hormone is abscisic acid. We have studied the three plant growth promoters, auxins, cytokinins and the gibberellins. Next we will go with the inhibitors hormones. So here around in the mid of 1960s, we can go with the year of 1960, the mid of 1960, three researchers reported three types of hormones which inhibited the growth or which stopped the growth. They were working on the plants and the hormones. They could find that there are three hormones or there are three chemicals secreted by the plants and this inhibitors will stop the growth but bring further development in the plants. So three different scientists or they published three different research were been submitted and they reported three different chemicals and those chemicals are inhibitor B, abscisic acid 2 and dominant. Three reporters, researchers reported three different chemicals, that is inhibitor B, abscisic acid 2 and dormant. These three were chemically studied and identified. When these were being identified, they were proved or they were being identified as identical chemicals, and this identical chemicals was named as abscisic acid. Abscisic acid. So this abscisic acid promoted, or it is a inhibitor hormone that stop the growth in the plants. So widely they are known as growth inhibitors, which inhibits plant growth and plant metabolism. This hormone was being identified as an inhibitor which inhibited the plant growth and plant metabolism, and these were also identified as those which were responsible for abscission and dormancy. Aging process, abscission, shedding of the leaves or the fruits and the flowers is known as abscission. Dormancy means the seeds became dormant, that is, does not germinate fast. The process of not germinating quickly or rapidly is known as dormancy. This chemical, abscisic acid, it induced abscission, that is the shedding of the flowers, fruits and the leaves, and dormancy. So it was being observed that it inhibited the growth of the plants and was being identified as a plant growth inhibitor. We'll go with the physiological roles. What are the physiological roles of abscisic acid? So this abscisic acid, as a growth inhibitor, inhibited the growth and also inhibited the physiological or the metabolic activities of the plants. So it is being identified as one of the hormone which promotes stomatal closure. It promotes stomatal closure. It is responsible, or it induces the stomatal closure. Stomata are the minute pores that are present on the leaves for the exchange of the gases. As we have studied in the anatomy class, that dicots and in monocots on the leaves, on the upper epidermis of the monocots, that is, they are present on both the epidermis of the leaves, so which help in exchange of gases, while in dicots the stomata are present on the lower epidermis, so we call it as hypostomatic leaves. So these stomatas are the minute cells or the organs which are present in the leaves, which help in exchange of the gases. So when there was secretion, when the plant secreted this abscisic acid, it promotes the stomatal closure. So when stomata get closed, partially closed, it will reduce the rate of transpiration, and this is very important in certain conditions, unfavorable conditions, where there is, like for example, there is cold, where there is shortage of water, or when there is scarcity of water. If there there is scarcity of water, so that the plant should survive for a longer period of time and to reduce the rate of transpiration, this acid stimulates the cells. It will help to stimulate the stomata to close. By the closure of the stomata, the rate of the transpiration is being reduced, especially when there is scarcity of water or during the cold seasons or in the cold regions. We can say that the stomata are being closed, by which the rate of respiration is being reduced. With the reduction of transpiration, water is being conserved in the plants for a longer period of time. So, if it promotes stomatal closure in the leaves, yes, where the stomata are being closed, and thus prevents or it reduces the rate of transpiration, prevents the shortage of water and reduces rate of transpiration. It reduces the rate of transpirations. Yes, the point is clear here. With the closure of the stomata, it initiates or it prevents the loss of water and prevents the problems of storage of water and also reduces the rate of the transpiration. So it promotes the stomatal closure and prevents the stress, the water loss. You can say it prevents the water loss and helps in storage of water and reduces the rate of transpiration. And it also inhibits, it induces seed germination. It induces the seed germination. So if you apply abscisic acid to such growing seeds or developing seeds, it will help to germinate very fast. It induces the process of germination in the seeds, which makes the seeds to germinate rapidly. That it is a rapid germination, or it will help to germinate the seeds very rapidly. Then it is one of the hormone. It is known as stress hormone. It is known as a stress hormone. As in human beings, adrenaline is being secreted, which is known as emergency hormone, which makes us to withstand the unfavorable conditions, or we can say that adrenaline is produced in the body in order to cope up with the situations. So there are certain stress conditions can be faced by the plants, like drought, floods or scarcity of water. During that stress conditions, this hormone is being released, which makes the plants to withstand the stress conditions. So it is a stress hormone which helps to face or withstand stress condition, to overcome the stress. So this hormone, as it is being secreted, as it is secreted in the plants, it makes the plants to face the situations, to withstand the conditions so that it can survive for a longer period of time. It will help to develop the plant, the tolerance. It makes the plants to tolerate to the conditions. So this is one of the stress hormone which helps to tolerate the difficult situations like scarcity of water or drought or flood conditions. So it is overcoming the stress. Even the plants also have the stress conditions. So these hormones help to overcome the stress conditions in the plants. And if you go with that, what are the other roles is, it is play an important role in seed development, maturation, and dormancy. So it plays an important role in the seed development, maturation, and also dormancy. Then if you go with the last point of the abscisic acid, it will help to withstand desiccation of the organs and other unfavorable factors. Then it will help to withstand the desiccation of the organs and other unfavorable factors. Apart from that, it works as an antagonist to gibberellins. So it will help to withstand desiccation, that is completely drying of the plant, of the organs, and also other unfavorable conditions. The main concept of this hormone is to protect the plants from the unfavorable conditions so that they can survive for a longer period of time. So this hormone will withstand, or it will prevent the desiccation of the leaves, complete drying of the part of the plants, and also other unfavorable conditions which may affect the growth or may affect the yield or the growth of the plants, and also works as an antagonist to the gibberellins. It is working against or you can say that it is the work of abscisic acid is opposite to the work of the gibberellins. So these are all the physiological roles of abscisic acid. Once again we will go with the points. As I said that it promotes stomatal closure. So when there are some conditions in the plants that are to be faced, when there is scarcity of the water, then the stomata is being closed, or it induces the stomata to close, which prevents, or it is helping to prevent the water loss and helps in storage of large amount of water by reducing the rate of the transpiration. So thus it promotes the stomatal closure, prevents the water loss and storage of water. The water is retained condition. It is one of a stress hormone. That stress hormones, this hormone, when it is produces, it will make the plants to tolerate the unfavorable conditions, or it will help to overcome those conditions or to overcome those situations. So it helps to overcome. It makes the plants to tolerate against the scarcity of water or high temperature, and even prevent the rate of transpiration. So it will help, as it is being secreted in the plants to tolerate the unfavorable conditions. It is called as stress hormone. It is secreted during stress conditions and makes the plant to tolerate. So it is called as a stress hormone. That it play an important role in seed development, maturation and dormancy. It helps to withstand desiccation, that is complete drying of the leaves. It will prevent the desiccation of the leaves or particular organs and other unfavorable conditions, while it works as an antagonist to the gibberellins. And the last hormone is ethylene, which is also an inhibitor. So in the tomorrow's class we will deal with ethylene, the inhibitors. So we have learnt the 3 promoters and one of the inhibitor, that is abscisic acid. In the next class we will go in detail the discovery of ethylene. So who did the discovery of the hormone? Cytokinins was discovered by Cook and Miller when they added the old extract of DNA. And one more point that I have to highlight is this kinetin was also being extracted from the hurling sperm. That is, it's a name of a fish. The DNA was being extracted from the fish sperm. Then it was treated in other plants, which promoted the growth. So this cytokinin promoted the growth, but opposite is abscisic acid, which inhibited the growth. So tomorrow's class we will deal with the hormone ethylene. Thank you.